Since the late 1980s, the Chum Technical Committee has been committed to the development and use of genetic characteristics to identify differences among stocks of chum salmon, differences that can be used and have been used for management and monitoring purposes. In this video, we discuss briefly the history of our development of a run reconstruction model, and specifically the exciting new results from a study conducted in the fall of 2016.
In our 2010 strategic plan, the Chum Technical Committee recognized the importance and the need for a run reconstruction model that incorporated both escapement and genetic information along with environmental variables. This Chum genetics environmental model was named ChumGem. When the design of the model was finished in 2012 at a spring meeting in LaConner, Washington, it was clear that a comprehensive genetic baseline was needed for the conceptualized model to work successfully. For several seasons, Chum Technical Committee facilitated the collection, analysis, and evaluation of an expanded baseline. Fortunately, through this work, the observed patterns of differentiation closely matched our units of chum management. It is clear from the dendrogram that Canadian and U.S. stocks are recognizably different. As well, different conservation units in Canada vary, as do stocks identified as management units in the U.S. These data have been and continue to be used by the Chum Technical Committee to monitor stocks entering Johnstone Straits, as well as in mixed stock fisheries in the San Juan Islands.
So we have this working version of the model, and we're able to see that it's very sensitive to certain parameters that drive it. One of those being what we call the diversion rate, which is the proportion of each stock that migrates on the outside versus the inside of Vancouver Island as they're returning to their rivers that are farther south than that. 2016, we were awarded some Southern Endowment Fund money to start a sampling program in the Strait of Juan de Fuca, collecting the chum salmon as they migrate through there. And so the objectives of this sampling program in the Strait of Juan de Fuca is to determine which stocks are migrating through there and get sort of a time distribution of when they arrive.
With resources from the Southern Fund, we were able to contract a purse-seined vessel to fish in the Strait of Juan de Fuca in both U.S. and Canadian waters over a 5-week period, starting the end of September through October.
So scales from the salmon that were caught in the test fishery were sent to the molecular genetics lab. We used 2 scales per fish to genotype 14 microsatellite loci. We had a success rate of just about 100%, about 99.5%, which is really good. It gave us a lot of data from the test fish samples. From that data, we were able to use genetics model developed by Pella and Masuda, which would assign the genotypes to different stock groups, and these were rolled up into management units. So for example, the results for week 40, broken up as Fraser, Southern BC, and U.S. Puget Sound. From the Canadian waters, the catch was proportionally greatest from the Fraser, very small amount of Puget Sound with a fairly large amount of Southern BC. In the U.S. waters, the catch was largely Fraser, a mix of Fraser, Puget Sound, and Southern BC as well. If we look at the Canadian side first, from week 40 on, the largest proportion was Fraser River in the red, with Southern BC, the next dominant group. A little bit of Puget Sound in week 41, but by week 42, Puget Sound, Southern BC, and Fraser were in equal proportions as the Fraser River proportion dropped. By week 45, the Fraser was a much smaller grouping overall. On the U.S. side, the Puget Sound group showed up right away in week 40, and that was maintained right through. So this would indicate that the U.S. fish heading towards Puget Sound would track along the U.S. shore rather than the Canadian side.
In summary, we feel this was a productive and successful study. We learned that a chum sampling program could be operated in the Straits across international waters, that stocks enter the Straits in different proportions, that apparently change through this time period, and that there are preferred locations for U.S. and Canadian stocks to enter through the Straits. We hope to be able to repeat this work to increase collection sizes, which we might do by sampling further into November. These are critical, timely baseline data that we hope to build upon to further our understanding of chum salmon abundance and population dynamics. Information that will help the PSC and user groups monitor, manage, and conserve this important and interesting species of Pacific salmon.
Very positive.
So as of 2015, we have a working beta version of the Chum Gem model, what am I saying? Hold on.